Good evening, everyone, and I heartily welcome you to the third edition of the African Science Frontiers Initiatives Seminar Series for 2022. For those who have been following us and so far, but if this is your first time, this is the second year we are running this seminar series. Last year, we focused on capacity building discussions. This year, our theme is spotlighting research that are advancing Africa's development. My name is Bright Waro, and I am the convener of this seminar series, and my co-convener is Dr. Eman Sob. Before I proceed, let me give Eman a minute to also introduce herself. Eman, over to you.
Welcome everyone to the second edition of the ASFI Science Frontiers Series 2022. We are pleased to have you all here and participate in discussions. I am Iman Sobh, Associate Professor of Respiratory Medicine at Al-Azhar University, Cairo, Egypt. And it's pleased me to be with you and to collaborate with you also. Thanks.
All right. So as you can see from the topic in front of you, it promises to be a very interesting and engaging discussion, marine biology, ion beam technologies, and nanotechnology, a journey into a multidisciplinary tango. Goodness me, it's full of some strange words and as well as very familiar words. So let's sit tight and see how this evening will go. I invite you, please, to really play your role by contributing to this discussion. Before I start, I have to give you somewhat bad news with regards to what we have this evening. Karen Cloete, who is speaking this evening, just got an emergency surgery to her eyes. And so that is painful, and it means that she is not able to be with us this evening. We didn't know this would come. It's an emergency, and we had to really pray for her to get well, but she was able to provide a recording of her presentation so that she will easily go through her surgery and recovery. Please, let's remember her in our prayers and encouragement. So what we are going to do this evening is that the session will still go ahead, and we will play the recording Karen has provided. I tell you, it's something that you wouldn't want to miss. So don't step out because of the news I've given to you. And then usually at the end of the speaker's presentation, we have question and answers. We'll still try to deal with that, but Karen has kindly also provided topics of discussion we can engage in after the presentation. Okay. All right. For those who are joining us for the first time, just to tell you what the African Science Frontiers Initiative is all about, and for those who have been here before, to remind you that our vision is to raise the next generation of African scientists who have the right competencies to drive Africa's developmental and transformational agenda through innovative scientific research. Our motivation is that Africa's development is not where we all aim it to be. And then, of course, we know that across every generation and continent and countries who have moved forward, science has always been a cardinal part of such stories. And so we think that the time has come for African science to be in the forefront of Africa's developmental discussion. For through what we do as scientists, the continent can have a leeway of transformation. So, doing that, it is very important that the capacity of the African scientists is built and up to date, and their competencies is up to the extent that they can use it to solve problems that the continent has. And so ASFI is here to contribute to the story of supporting African scientists to build their capacities and competencies in doing their research. Our mission is to instill excellence. For those who have been attending our courses, our programs, you will realize that we have taken this very seriously to instill excellence through competence acquisition, capacity building, and career development. We want to contribute to Africa's science that tackles Africa's developmental challenges. We believe that only a competent science can change the current narrative of the continent. Our activities include seminars and workshops as we have here this evening. We have hands-on courses in different areas of the scientific research methods, capacity building and career development, science leadership, science communication. We have various mentoring programs in which we support early career scientists as well as senior ones to really enhance their capacities to do what they're doing in a hands-on manner. We also have research programs in which we try to investigate some of the challenges that the African scientists are facing, to highlight this topic either through our seminars or develop courses that we can train scientists to overcome those challenges. We started just over 1.5 years ago now, and then we have implemented 7 courses, each of them running for 5 days, each day about 8 hours. We have received over 3,200 applications, and which we have trained 817 participants during this period. And in total, the participants have come from around 40 countries in Africa. So ASFI is making wave across different countries. And one thing is that we're happy to note that participants have appreciated what we offer in this platform. As I mentioned, last year we had a seminar series which theme was Disrupting Stagnation, Empowering Africa's Future. We brought different speakers from different perspectives with the goal of presenting topics that we challenge African scientists to build their career programs. And so, yeah, we also had other aspects like the My Career, My Story, which was a quarterly program to complement the seminar series in which we invited Africans across board to share their career stories in a way that can motivate African scientists, fellow scientists. And all we do, our courses and the seminar series are freely available on our YouTube channel. So please, If you have missed any session, if you have missed any of our trainings, you have the freedom to go to our YouTube channel and tap into the resources we have there. This year, we continue the seminar series, and we're inviting people from different fields of endeavor, with different fields of research, to share their experience on what they are doing and what they are doing in which way that is contributing to Africa's development. We also have the tips and tricks, research tips and tricks, which we have had the 3rd edition last Friday, and that is one of the workshops we run. The 4th edition is coming on 20th of May. So please feel free to join us in these programs. All right, today's moderator is Mr. Farai Kevin Munyai. Kevin is currently based in South Africa, but he's originally from Zimbabwe. He is a public health professional and biomedical scientist or practitioner. Currently is a PhD candidate at the University of Western Cape, South Africa. He had also his master's in public health from the same university, Bachelor's of Medical Laboratory Sciences from the University of Zimbabwe. Kevin has had lots of experiences working with different organizations in the area of health technology, public health, health policy, and so he's quite experienced, motivated in what he's doing, and has been part of the ASFI alumni and a participant in our programs. So it is my honor to introduce to you Mr. Kevin, who will be moderating this session this evening. So Kevin, it's over to you, and thank you for accepting this responsibility this evening.
Thank you, Prof. Bright. I hope you can hear me loud and clear.
Yes, we hear you clearly.
Okay. Thank you so much, Prof. Bright, for such a humbling introduction. Sometimes when someone introduces you in such a way, you wonder if they're really talking about you or somebody else. So thank you so much for that. Thank you for considering me for this great opportunity to participate on this great platform to advance this science revolution and driving the African continent's developmental and transformational agenda. So it is a great pleasure, it's a great honor to be part of this initiative. Thank you for this opportunity. Back to you.
All right, I think you can go straight, Kevin, to introduce the speaker and our topic for this evening.
Right, thank you. So it is now my pleasure to introduce the topic. I think Prof. Bright shared with the group earlier on. The topic for our talk tonight or today, it might be morning in some of the areas where you are connecting from, it might be evening. I'm currently in Namibia. As Prof. Bright said, I'm a student in South Africa at the South African University doing my PhD in public health. So currently in Namibia, it is evening time. So our topic for today is marrying biology, ion beam technologies, and nanotechnology, a journey into a multidisciplinary tango. I attended a session, I think last week. We had a course that we are attending, and one of the participants said, you know, the topic is so interesting, it sounds like some kind of a relationship which is not only two-way, maybe three-way kind of relationship. So there were people who really had interesting comments about this topic. And it's really interesting for me as someone who has been in the biology, medicine, laboratory medicine space. These new topics that are coming up, new technologies that are coming up, they are really quite interesting. And looking into the future to see what this brings into science and into the development agenda for our continent. So I'd like to introduce our speaker for tonight, Dr. Karen Cloete. She is a research fellow affiliated to the UNESCO University of South Africa Chair in Nanosciences and Nanotechnology, Nanosciences, African Network, iThemba Labs, the National Research Foundation, and her research focuses on chemical fingerprinting of biological material with ion beam and synchrotron radiation technologies for application in diverse fields. And currently she is working within the field of nanosciences and nanotechnology and its applications. She has been the recipient of various scholarships and grants and awards, and she has published interdisciplinary research papers, and her work has been highlighted in popular media and at international academic and non-academic fora. She has attended many conferences, and she has developed and lectured various courses for international and national institutions, and has mentored and co-supervised national and international fellows from interdisciplinary fields. She has also contributed to the formulation of statements on topical issues in science, and has served on various committees and as co-lead of different working groups, as well as served as a consultant for national agricultural companies. She strongly believes in the power of science to transform society. She has also been a guest on one of the ASFI series, where she also shared some of her experiences in the academia and also in the industry. So those who are interested in looking more into her work, there is a recording that is available on the ASFI channel on YouTube. All right. So I myself have not had much experience in nanotechnology or ion beam science. It's something that I've had in the corridors at school or at universities. So I'm really interested in hearing what is going to be shared in this session. One of my colleagues, when I shared the flyer, they were asking me what this is all about. And fortunately, there was someone nearby, and then they said an example of what a nanometer is. If you look at human hair, if there was a hair for the hair, that's how small the nanoparticles really are. So this would be also another learning opportunity for me. I've been in the medical field for a while, and there are issues around diagnostics that are being developed to really use these new technologies, these advancements in technology to help with diagnostics. There are also examples of drug delivery mechanisms that can use nanotechnology. And also another key element where we can use nanotechnology includes also cancer treatments, and there are quite a number of researches going into that, and including also brain studies. So this is quite an interesting area for me, and I can't wait to really listen to the session that Dr. Karen Cloete has provided for us. For those who are just joining, it is quite unfortunate that our speaker for this session could not be with us tonight, but it is great that she has managed to provide a recording for us to go ahead with this session. So we'll be listening to the recording, and then at the end of the session, we'll have a chance to have some discussions. She has also kindly provided some discussion questions for the participants for us to have a deeper discussion and understanding of what the link between the biology, ion beam technology, and nanotechnology can bring in terms of our scientific developments. So, Prof. Bright, if you could kindly share the recording, and then we can have our discussion after the recording is finished. Thank you.
All right. Do you see sharing?
Yes, it is up on the screen.
It's fine.
Okay, since you're not hearing it yet, just give me a minute.
Good evening, esteemed colleagues, senior and junior fellows. I want to extend to you a very, very warm welcome for making the time to attend my talk tonight. Thank you very much. So the title of my talk tonight is Marrying Biology, Ion Beam Technologies, and Nanotechnology. My name is Cloete, and please feel most welcome if you have any questions or would like to get in touch with me to email me at the address provided. Before I start my talk, I would like to extend a very warm thank you to Prof. Bright for inviting me and running this wonderful initiative. Good evening, esteemed colleagues, senior and junior fellows. I want to extend to you a very, very warm welcome for making the time to attend my talk tonight. Thank you very much. So the title of my talk tonight is Marrying Biology, Ion Beam Technologies, and Nanotechnology. My name is Cloete, and please feel most welcome if you have any questions or would like to get in touch with me to email me at the address provided. Before I start my talk, I would like to extend a very warm thank you to Prof. Bright for inviting me and running this wonderful initiative. So, again, before I start, I would like to provide you with a brief overview of the outlay of my talk that I'll be discussing with you tonight. So, I'm going to start by discussing my personal journey with you.
Yeah, so the video has stopped playing.
I will try it again. Sorry.
Oh, okay.
Let me try it. I think. Yeah.
Good evening, esteemed colleagues, senior and junior fellows. I want to extend to you a very, very warm welcome for making the time to attend.
Do you get it now?
Yes, right.
Yes,
You see
it is playing okay now. Yeah.
Okay.
My talk tonight. Thank you very much. So the title of my talk tonight is Marrying Biology, Ion Beam Technologies, and Nanotechnology. My name is Karen Cloete, and please feel most welcome if you have any questions or would like to get in touch with me to email me at the address provided. Before I start my talk, I would like to extend a very warm thank you to Prof. Bright for inviting me and running this wonderful initiative. So, again, before I start, I would like to provide you with a brief overview of the outlay of my talk that I'll be discussing with you tonight. So, I'm going to start by discussing my personal journey with you. Then I'm going to focus more on collaborations and why it is so important for young and early career researchers to collaborate with colleagues in their local country and also abroad. Then I'm going to focus a bit on, just very briefly, the considerations that shaped my research journey and why this is so important for young scientists to carefully plan their research journey ahead. Then I'm going to focus on my research, all of the different research that I did most recently and also currently I'm doing, and then finalize with acknowledgements. So many years ago, I started as a PhD student in microbiology at the University of Stellenbosch. I was very interested in microbiology because it was such a, microbiology is such a diverse field. You can apply it to food microbiology, to medical microbiology, to agriculture. So it's a very diverse and very interesting field. So I thought this would be a good idea to pursue this because I can really diversify my research portfolio. Now, when I was busy during my PhD studies, one of my co-supervisors exposed me to a technique called Pixie. And the full name of the technique is proton-induced X-ray emission. Now, this is an elemental mapping technique. So you expose samples to a beam containing specific types of particles. These particles interact with your sample, and then you obtain maps of different elements that occur in your specific specimen. And I found this technique really fascinating because it can be applied in so many diverse fields, in biology, in material science, in nanosciences. And I decided to do a postdoc at iThemba Labs. So iThemba Labs, the full term stands for iThemba Laboratory for Accelerator-Based Sciences. And this is one of the National Research Foundation of South Africa's research institutes here in Cape Town. So I spent a couple of years there. And then I was welcomed very warmly into the amazing group of Prof. Maza, who is the UNESCO-UNISA Africa Chair in Nanosciences and Nanotechnologies. And this is a joint position based at UNISA, the University of South Africa, and also at iThemba Labs in Cape Town. So for those of you that haven't visited South Africa yet, here's just a picture of Cape Town and beautiful Table Mountain, and iThemba Labs is very close to Cape Town. Right, so let's start by giving you also a brief overview of my collaboration. So throughout my career to date, I've established a lot of different collaborations with colleagues in different disciplines in Africa, in the UK, in Europe, in Russia, in Jamaica, and Pakistan. And why is this so important? Especially when you're a young scientist, when you attend conferences, mark off the speakers that you are interested to meet and arrange meetings with them, exchange your data with them, pass your articles to them, because these are good opportunities to develop links with other researchers. And this is very important because that exposes you to funding opportunities, right? To world-class knowledge and also to world-class instrumentation. And that builds the impact of your research. If you have nice and good, robust collaborations, a lot of opportunities become available to you. You learn as a young scientist. You grow as a human being because you interact with so many different and interesting persons. So think about this. When you plan your career, also carefully plan your collaborations. Right. What are the considerations that shaped my research journey? So when you're a young scientist, don't just think about finishing your PhD. Think about planning your journey, where you see yourself in 10 years. And now answering a basic research question is not that fundamental anymore. It is, but one needs to think of outside of the box. Research is diversifying a lot. We need to think about doing multidisciplinary research. We need to think about addressing the sustainable development goals. The world is facing currently a lot of grand challenges. And to answer those very worrying questions, one needs to collaborate, first of all, and one needs to think about performing or combining fundamental science with applied science. So if we are studying a system, we need to fundamentally understand it. But then we need to take that knowledge also and apply it to assist, to guide, to help society. And that is where also multidisciplinary science come in, because the problems that we are facing today are so complex. We need to collaborate outside of our immediate silo. And think about if we're in, for example, in microbiology, how can we apply microbiology, for example, in nanotechnology? Or vice versa. How can we apply microbiology in agriculture? Instead of just zooming in and working in our silos, we need to extend beyond that, reach beyond that to answer these very, very complex questions while still having your main thread of research focus. Then very important also, think about trust in science. Nowadays, the public trust in science are starting to waver, and that is why communication outside of traditional academia is very important. Also, besides publishing in purely academic journals, try and communicate your work to the general public. tell them why science is so important. What are you doing? What are you doing that can help or guide society? And a lot of the things that we are doing as scientists can be applied in the real world, but we are so focused on being within our academic silos, and sometimes it gets so busy within that silo that we forget to also reach out to the public. So now, besides planning your research journey, thinking about your path, your collaborations, whether you want to perform fundamental and applied research, do multidisciplinary research, think about, there's so many things to think about that. And for that, I would also recommend reading good online materials. There's a good book by Elsevier called Early Career Guidance for Young Fellows, if I can remember correctly. But read resources that are available on the internet to guide your career. Early career guidance, there's a lot available on Nature, on Elsevier websites. I know that Nature also has a careers section where there's a lot of useful articles about career guidance. But now let's focus on the research. So to date, I've been involved in a lot of multidisciplinary research areas. For example, here, what you see on this slide, it's a bit busy, but let me slowly go through this. So at one stage, I thought it would be very interesting to work on human samples. And performing work on human samples can be very interesting, but one needs to remember all the other aspects surrounding that as well. For example, one needs ethical clearance. That's one aspect. So for example, here we worked on hair. We worked on human hair. We took some samples of scalp hair, and we wanted to look at the elemental profile of that using Pixie. And we performed point analysis to look at the elements in hair. Why is this so important? Again, thinking about the multidisciplinary aspect. One, hair is one of the most robust substances to work on if one looks at environmental exposures to pollutants. Nowadays, our environments are very much polluted. We are exposed to a lot of toxic metals. For example, India and Pakistan has a huge problem with arsenic. Hair can be used to test and screen for arsenic. Why? Because it's a very stable matrix. The hair preserves these elements because of its unique chemical composition. So it's a useful tool in environmental toxicology. Then another technique that we used was this secondary ion mass spectrometry, and this is linked to, this is not a benchtop technique, you get the benchtop one as well, but this can also be linked to an accelerator where you use ions to irradiate the hair and then get a specific profile of your hair in terms of its organic and also elemental profile. And what was interesting, that what we saw in the hair using this technique is that our hair contained lithium. Now, again, if we think in a multidisciplinary fashion, lithium in hair is important because one, it can be used for biomedical applications. Why am I saying this? In bipolar patients, they are given lithium medication. Now, sometimes in such populations, they don't adhere to their medication regimen. And this can be used to test or screen together with other techniques if a patient is adhering to their medication. There can be flaws in the study design as well. That's why I'm saying careful collaboration with a diverse team is important in this aspect. Then secondly, what is very useful is one can obtain a map with both Pixie. If you use Pixie for elemental analysis and the SIMS also, you can get a nice map across your hair length. And this provides you with a time profile of what you are looking for in your hair, whether it be an element or whether it be an organic compound, for example, like a drug. If you want to screen for drug use, these are nice techniques to use. Then this Daphne is a very interesting technique because you can pinpoint a specific area in your hair. For example, if you focus on the medulla, this region here, it points more to a substance that have been taken by the mouth, ingested. Whereas if you screen the outside of the hair, it can point to substances deposited on your hair. So for example, there's a lot of false screening tests in drug studies when someone may be exposed to the smoke of a drug and then test positive while they were not actually taking the drug. So when we look at this type of research on hair using these set of techniques, again, what I mentioned earlier, if one thinks about it in a multidisciplinary fashion, you can apply this to forensics to test for drugs. You can apply this into environmental toxicology to test for pollutants. And again, one can test it in biomedical studies. So that was one part of the research that we looked at. And I'll focus in a bit more detail on that a bit later. Then at a later stage, we started to focus on applying Pixie and other techniques like CT scanning. This is a CT scanning image that you see here, micro CT more specifically, to look at, we used also a multidisciplinary approach to food science to look at the porosity and also the elemental profile of coffee. And if we think about that also in a multidisciplinary fashion, one can also apply that to forensics, also to environmental toxicology, that I'll also focus a bit on later. So this is more plant-based samples. Then we looked also at a study that focused on studying the interaction between soil microorganisms and plants. And then also very interestingly at plants that accumulate metals and clean up the environment in a method called phytoremediation. So let's look at some of the first studies that we recently performed. So these studies are mostly within the field of microbiology and botany, and more specifically focusing on biofertilizers. So this study focused on analyzing a South African plant. This plant grows in the fynbos biome. It's a very, very popular herbal medicinal plant. It is called Agathosma betulina. It's buchu, so it's well known for its very highly aromatic oils. Also, that is used in the cosmetic industry, in perfumes, in also beauty products, also for its medicinal value, especially drinking this type of plant's water is very good to clean your kidneys, right? But establishing this plant as an agricultural crop is very, very difficult because it grows in poor nutrient soils. It struggles to establish itself. So we were pondering, what could there be in soil that could assist in guiding this plant to promote its growth performance? What could there be in soil that could boost the plant to grow faster, to grow better, to take up more nutrients? Now, if we look at the soil here, we think about soil in a multidisciplinary way. Soil contains bacteria, it contains nutrients, and it contains different insects, for example. But we were specifically looking at yeast, soil yeast, and we were trying to isolate the soil yeast, then inoculate it onto the plant and see how it affects growth performance of the plant, and more specifically, nutrient transfer. So as I mentioned earlier in my journey, we were looking for a specific technique that we could use to study what was the relationship between this yeast and the plant, and is it transferring useful nutrients to the plant, or, just in more better detail, understand the relationship between this yeast and the plant and the nutrient transfer between these two organisms. Because if you have good nutrient transfer, for example, the yeast dissolves poorly available nutrients and transfers this to the plant. The yeast can serve as a very good biofertilizer to promote the plant's growth for agricultural purposes. So now we looked at different techniques that could be used to study this interaction. Now this, what we specifically wanted to assess was nutrient levels within the plant. But when you choose a technique, remember all the techniques has their pros and their cons. So one has to think about this very carefully. Also using a combination of techniques is also very good, right? One must think about sample preparation, for example, because when one looks at elements in a plant, if you bulk process any type of material, there is the possibility of introducing contaminants. Also, you destroy the sample. You cannot do microscopy studies on the sample itself to see the location of where the yeast is growing, or for example, you cannot locate where a specific element is located in the plant, because bulk processing gives you eventually at the end of the day a powder, right? Also, the material that you use to process any type of sample depends on the type of equipment you use, right? Also on the characteristics of the material itself, where the material is hard, its chemical composition, plasticity of the sample. Also, when you process, you can use volatile elements that may be important, for example, light elements like lithium. Then also the type of material you use to process your sample can also influence the eventual output when you are specifically looking at elements. So why did we decide on using Pixie? Remember I mentioned earlier that Pixie is an elemental analytical technique that where you can, when you analyze your sample, you obtain a map, and you can get a map of each element across the periodic table higher than sodium. You get a specific map providing you with quantitative information about the distribution of that element in your sample. Most importantly, the technique is non-destructive. So you prepare your sample in a certain way that is still intact, and you can obtain a map from different morphological regions. Whereas when you use bulk processing technique, you have to grind a sample or process it with chemicals that can introduce toxins or, foreign elements, contaminants, and also you don't have an intact sample anymore. For some Pixie techniques, virtually no sample preparation is required. It all depends on whether you're working on vacuum or not. You don't have much damage after sample analysis, depending on your sample type. And also, it's a highly sensitive technique, and it's fully quantitative. So let's look at the setup of the Pixie technique. Remember, it's similar to other elemental techniques, but it can provide a map. So it's similar to ICP, MS, atomic absorption, spectroscopy, but the setup of it is a bit more complicated. So what we have here is, this is your sample chamber. So this is where your sample will be located. Your sample is irradiated with protons that interact with your sample, that I'll explain a bit later, and gives you eventually down the line your elemental map that you need. And this is your detector here, right? So this image is actually here at the end station. So here you have your accelerator system that creates your protons or your particles that travels down here, and there they interact with your sample to provide you at the end of the road with your elemental map. So in the system here, the tandem accelerator, you get ions with a certain energy that are extracted from an ion source that is here at the back. They are analyzed in here, and they are injected into the system here where they are accelerated, right? First, the injected negative ions are accelerated due to applied electrostatic forces, right? And in the terminal end, the injected negative ions are stripped, and then eventually you'll get your desired particle traveling down this line. For Pixie, one uses traditionally protons. So this is just a very, very brief and very simplistic overview of the setup of this type of system. You have the accelerator producing the particles, traveling down, interacting with your sample, and providing you, at the end of the day, with your result. This is an image of what your sample looks like. And what you're seeing here is a root section. So this root section was inoculated with yeast cells. And we wanted to see, does the yeast transfer nutrients to within the root, and where are the nutrients located, right? And this is the sample holder that is placed in the sample chamber here. So if we look at the principle of the Pixie technique, our sample is constituted, or elements rather in your sample is constituted of different types of atoms, right? And when the beam interacts with your sample, the interaction on a more, on a more molecular level, when that happens, an electron is ejected from the inner shell of an atom of an element within your sample, right? So an electron from a higher shell drops into this lower shell to fill the vacancy left there. When this happens, an X-ray is emitted, equal in energy to the energy difference between the two shells. So that is the basic principle on which this technique is based, right? So let's look at the type of data you obtain when analyzing a sample or a specimen. It can be biological, it can be material sample with this type of technique. So for example, here we see data obtained from a root section originating from a plant that was inoculated with a soil yeast. And here you can see quantitative data from the entire root area and from certain areas within the root tissue. And one can clearly see here that the yeast, this is the yeast inoculated and this is the control, that the yeast has a significant influence on pea nutrition, right? So one can see clearly using this technique that in this interaction, in this symbiosis, the yeast is transferring nutrients to the plant. What we also obtained with this and that type of analysis was an elemental map. And here we can see, because remember, when we also performed microscopy analysis. When one is doing research, when you have a research question to answer, one often needs to use a whole suite of techniques. But the use of those techniques will depend, again, on your main research question. It needs to assist you and guide you in answering that specific question. So with the microscopy, we saw that the yeast, it colonizes the outer regions of the plant. And with the Pixie, we could see that the phosphorus was mainly concentrated in these outer regions of your root, clearly showing that this microorganism that occurs in the soil is contributing to the nutrition of the plant. And what was also interesting to see with other techniques is the plant preserves this phosphorus in these outer regions. And during periods of pure availability, or when the plant really needs it, it releases this phosphorus and makes it available to the plant. Same can be seen here for iron. The yeast also assists the plant in preserving iron in the outer regions, right? So with this technique, you can see an elemental map, And you can also see the distribution of where the element is located at a quantitative level. Right? As I said earlier, it's always good to think about applying a whole suite of different techniques when you want to answer a research question. For example, here, as I mentioned, we could see with fluorescence microscopy, when we use the specific dye to stain the microbial inoculated roots, we could see that here is live yeasts growing on the surface of the root, responsible for transferring this phosphorus, dissolving it in the soil, and assisting it in transferring it to the plant. Here, it's also important when you look, for example, at such elemental maps to do also microscopy imaging so that you can relate your elemental map to a morphological image to more clearly assess where a specific element is located, for example, in what morphological region. And what we also found was that in the outer regions of the root, there are these bands called Casparian bands that almost acts like a locking system, locking in nutrients in a specific region. So, right, the conclusion of the study eventually was that yeast can significantly affect host nutrition, and it can impact plant fitness in the absence of mycorrhizal fungi. Now, mycorrhizal fungi is also a type of microorganism that occurs in soil, and also this is a very popular type of biofertilizer used in agriculture. There's already a range of agricultural companies selling mycorrhizal fungi to farmers to promote plant growth. So, in sum, this study used a suite of different techniques, a very advanced elemental mapping technique to really understand on a fundamental level, to apply this research in agriculture, what is the relationship between yeast and plants, yeast specifically that occur in the soil or that occur naturally in the soil, and plants, and that can be used as biofertilizers applied in agriculture as a fertilizer to boost plant growth. Right, let's move now to the next study. Right, this is still within the field of botany, and here we look specifically at metal accumulators and to quantitatively assess where elements that are taken up by plants that grow in very highly polluted areas, where does these metals go? Where do the plants store them? Now, this is both a fundamental study to understand where toxic metals are stored within the plant, and also it can be applied. Why? Because such plants, they grow in highly polluted areas, and they have the unique capability to take up high levels of, for example, copper, nickel, even arsenic, very toxic elements. They take up in high volumes in their different plant organs, and they process them there, and they can effectively survive in this environment. Now, such plants can be used to locate where metals are located in specific areas. For example, is this area high in copper? It can also be used to phytoremediate contaminated areas. For example, by planting this type of plants on the area, it's a natural cleaning up process. The plant takes up the toxic elements, and then one can harvest that again for biomining. For example, if the plant now stores these toxic metals in the leaf, one can harvest them, and then you can use that in biomining. So this study specifically looked at plants that occur in Botswana. Now, these plants are called Metallophytes, meaning they take up metals as part of their natural physiological process. And why we wanted to see, where are these metals located? And again, Pixie provided us with a good overview about where in the leaf you get the highest concentration of elements. And this is a transverse line graph that shows you also the distribution of the elements across the different leaf regions, right? Also, it was possible to obtain an elemental map of the leaf. Here is the microscope image of the leaf, and here is the Pixie elemental map. For example, here we can see where copper is located. When we compare this map with a microscope image, one can clearly see that some of the copper is deposited here in the epidermal leaf regions, right? So let us conclude. The difference in concentration and accumulation patterns between these two plants suggests that the metal-tolerant adaptation of these plants may differ, right? So one will maybe accumulate one metal more than the other, and this has very important implications for the applied aspect of this study, of this type of studies on metallophytes, right? So let's now move on to the last study, I think. So as I explained earlier, hair is a very, very useful substance to look at, or material to look at, and it can be applied in very, very diverse fields. For example, in forensic science, if we now specifically focus on forensic science. Now, I know in South Africa, there's a huge problem with identifying unidentified remains of bodies found in open fields. It's very difficult to link the evidence to a specific person, and forensic science here is a huge, huge field of great importance in solving crime, right? So often only physical evidence is available on a crime site. For example, hair, blood, nails, something like this. When we have intercontact between a victim and a perpetrator in a violent crime, hair is often transferred between these two parties, right? It can easily be seen, it can easily be studied, because it provides microscopic and both microscopic evidence. For example, one can distinguish between an animal versus a human hair, right? And it's a very useful substance because it has a very high keratin content and is resistant to decay, unlike blood and other types of samples. It stores a higher concentration of chemical compounds, for example, drugs, in the hair. It has a longer diagnostic window depending on its length. You can do retrospective profiling. And it can also be collected under close supervision. For example, if you want to perform hair screening to assess medication adherence. And obviously, it's easier to collect, to transport, and less invasive than drawing blood. And then again, hair can be used very, very widely in therapeutic monitoring, medicinal versus drug use, for gestational drug use to test that. And most importantly, where hair is very widely used is to assess for exposure to environmental toxins. Right. So let's look at the scope of different studies we have already prepared on hair. So this was a specific study focused on Anatolian shepherd dogs. And these dogs are very widely used in South Africa to guard livestock from predators. Instead of using traps or killing predators, you use this dog to safeguard your livestock with minimal impact on the environment, right? It is used for that. And this study was specifically looking at analyzing the feces obtained from farms using these type of dogs to identify the prey species, right? Whether the dogs prey on the livestock that they are supposed to protect, or whether they are other wildlife preying on the livestock, and also to identify the livestock that is being preyed on. Now, right, if we look at animal hair, different species, right? This can be used hair characteristics, like for example, the anatomy of the medulla and also the morphology of the scales can be used to identify different types of animal species. Each type of animal species has a very unique hair pattern, and that can be used to identify whether to answer. If you look at the feces of these dogs or other feces available and collected from these farms, what kind of prey, what kind of livestock are being preyed on, or what general kind of wildlife are being preyed on by the animals that freely roam there. So that was one study, and here we could link the specific hair that we sampled to a specific animal species, right? So let's move on here to the elemental studies of the hair that we specifically focused on environmental toxicology and also to drug analysis. So this is a picture of the hair that was analyzed with Pixie. This is a sample holder for Pixie, and this is how the hair was mounted. This is an image of how the hair was mounted for the SIMS, the secondary ion mass spectrometer. Remember, this Pixie technique is for elements, and this technique is mostly focused on analyzing organic compounds. So you can see the distribution of elements across your sample and also of organic molecules such as drugs, for example, across the length of your hair. And remember what I said previously, you don't need chemical processing. You preserve the internal and external morphology of your hair. So you can look at, you can screen different regions of the hair. For example, if you find that an element is located here inside the medulla, it's more likely that the person ingested it. If the element or organic compound is found here, it's more likely that it will be because of aerial exposure, right? And then with the Daphne technique, you can also zoom in a specific area, for example, the medulla, to extract specific compounds, for example, drugs from that specific region that links or points to ingestion. Right, so I just here provided a very brief overview of the SIMS technique. So a primary ion pulse is focused and scanned over your sample system. Remember, your sample is under high vacuum, similar to Pixie. But with Pixie, you also get the option of performing Pixie in air that does not use vacuum. And remember, when you are analyzing a sample in vacuum, the sample processing for that is extremely important. If you don't process your sample correctly, it may influence your results. That is what I mentioned earlier. When you are choosing a technique, you need to understand how to prepare your sample for it, and also what is the pros and cons of that specific techniques. So then coming back to the SIMS technique now, secondary ions are released by the impact of the mega electron volt, right, providing primary particles. The yield of these secondary ions depends again on the incident angle, the energy of the beam. So there's a lot of different factors of your experimental setup that will influence the yield of your ions. Then the drift velocity of the secondary ions reach then the detector, and this depends on the mass to charge ratio, and the technique allows for mass spectra to be extracted from an image and vice versa. So this is the experimental setup of the SIMS technique. It's very similar to Pixie. Here you have your beam, particles traveling down the beam. Here you will have your sample setup, and here you will have your detector. And what is mentioned here is in certain biological samples, sometimes it's preferred to analyze a biological sample with Pixie under cryo conditions. So the cryo conditions preserves the very, very natural state of your sample. It's closest to its natural state. You analyze it under liquid nitrogen conditions. You flash freeze your sample, and then you analyze it also under liquid nitrogen. And this is provided here by this setup. If such a Pixie setup has such a system, you can analyze your sample under cryo conditions, and then you can analyze it closest to its natural state. Right. So if we again look at the data, what we obtained using this combination of different techniques, you can get elements from here that can be linked again with environmental toxicology. You can obtain information about the organic compounds, drugs that a potential suspect is using, and then you can pinpoint whether the drug was ingested or not. And this is a type of the type of data you obtain with a Pixie. As I mentioned before, you can get a spectra, you get quantitative values, and also you get a quantitative elemental map. And here we can see where the sample was analyzed with the Pixie. That's why it's so important to also take microscopic images of your sample to see if the Pixie setup damaged your sample, because here we can see that this region cannot be used for subsequent analysis, additional analysis, because it's already a bit damaged here. Then with the MEV SIMS, we could detect various pharmaceutical compounds, drugs, and also its distribution across the different hair morphological regions. And what was also very interesting was we detected lithium isotopes in hair that could point to either use of medication or alternatively for cosmetic applications, since some of the hair products that are used by women mostly contain lithium. Right. Now with Daphne, it was very interesting to see this sample here was obtained from a male Italian prisoner. And this really shows the importance of where in the hair one takes a sample from. Because here we can see clear difference in the peak height of cocaine that was detected in these different regions of the hair, right? So let's come to the conclusion. When we did the study that I mentioned first on the animal hair, we also tested different sectioning techniques. So we combined the study focused on different microscopy and also different sectioning techniques. Now, when one thinks about it, how you section a hair can also influence your end results. If you perform bad sectioning using a bad technique, you will at the end get a bad technique. And when I go back to these images here, if we focus on the technique that we used for sectioning to look at the morphology of the hair to identify the species, this was a hand section, and this was cut, these two here was cut with a microtome. And here one can clearly see the difference in sectioning can have on your sample characteristics. Also the level of detail you can see with microscopy to perform analysis of what species you are dealing with. So let's get back to this last study here. Right. So that was the conclusion for Pixie. Let's look now at the conclusion of the Pixie results. So what we also obtained in some of our hair samples that we analyzed is some of the hair samples had high levels of chlorine and bromium. Now this again can originate from various sources, right? It's also important that if you link it with the morphological properties of hair, thinking about it in a multidisciplinary fashion, is chlorine can damage chemical properties of your hair. It can cause hair damage. So if you are exposed to a lot of chlorine, it can eventually cause hair damage because it can diffuse into your hair shaft where it reacts with melanin and lipids, right? And also, what also influences hair analysis, and that sometimes it's not always focused on and highlighted in forensic studies, is the levels of melanin in hair. We found that hair with more melanin had a higher metal content, right? So remember what I said about thinking in a multidisciplinary way. It's important to correctly answer different types of research questions. Also collaborating, if you are, for example, focusing on elemental analysis, it's also important to collaborate with researchers who are solely focused on, for example, cosmetic type of analysis, working on hair, working on skin, etc., to establish those links. Because if you get a result for a specific element, and it can be because of various reasons. It can be environmental exposures. It can be due to the chemistry of the hair itself. So then what was very interesting also, we analyzed different types of hair, and we found that also in hair with higher melanin, so darker hair, and also hair that are more curly, have a higher calcium content, right? If one links that to the hair chemistry, is calcium is used in the production of keratinized proteins, but it can also originate from soaps, etc. So one needs to also think about that, consider that when one is doing forensic research. When one hair has more calcium than the other, could it be that the hair's curliness or morphological parameters influence that elemental level, right? Then I've already spoken a little bit about the lithium, and then the advantages of using all three techniques. Now, if we move on to food analysis. So there was also another study that we were performing, and we were looking at specifically coffee. Now, coffee is a very popular and desired beverage. It's used for its obviously hedonistic, psychostimulant, health benefits, pharmaceutical, cosmetic, and coffee has a very unique elemental structure or signature that is also linked to its quality. So there are various factors that can influence the elemental levels in your coffee, if you think about it. For example, roasting. Also, the introduction of leaves and other contaminants during processing. Also, the bean variety and the region where it comes from. For example, coffee from a specific region can have a very unique elemental profile. And this can again be linked to forensics of food studies. For example, nowadays, a lot of food fraud is happening, right? And this type of screening can then be used to link if a certain food sample is originating from a specific region. But again, these things, elements, and other, for example, characteristics of a coffee can be linked to environmental conditions, soil conditions, also farming practices, where the farmers are using different insecticides, whether they're using organic or conventional farming styles. So again, thinking broadly about the topic is very, very important. So in this study, we wanted to screen the elemental profile of different coffees coming from different regions, to try and see if elemental composition can be linked to a specific region, and also, again, extending that to food fraud studies. And then we also wanted to combine Pixie with another technique to assess the quality of the roasted coffee and how roasting affects porosity, because if you have a more porous product, it can be more aromatic. So we obtained some elemental maps. We saw that the elemental profile of different coffees from different regions is very unique, as depicted here in this heat map here. Certain regions clustered together, certain elements also clustered together that can point to different explanations like soil profile, farming profile. And then we could also see here that coffees from different regions also exhibited a different porosity profile that can be linked to its quality, right? So today I'm finding myself within the field of nanotechnology, and specifically the biological applications of nanotechnologies and nanosciences. So what we're currently focusing on our group is synthesizing nanoparticles using a diverse set of techniques. For myself, I am using a green chemistry, and by that I'm using green materials, for example, plants, that I use as a starting material to synthesize nanoparticles that I apply in different research fields. Now, the benefit of synthesizing nanoparticles using natural products, like, for example, plants as a reducing agent, is that it produces nanoparticles with a very unique physicochemical profile, right? Secondly, it's safer. It's less environmentally toxic if you're going to apply it, for example, in agriculture. Now, what we're also currently looking at is we want to develop a nanofertilizer. So currently, within the fertilizer industry, a lot of issues are being experienced. We have climate change. We have the Ukrainian war that is also significantly affecting fertilizer supply. And current fertilizers are also not effective enough to enhance nutrient use efficiency in plants. Farmers sometimes oversupply plants with fertilizers that become unavailable, they become washed away, they become captured or accumulated in the soil environment, and they may not become available to plants to effectively use for their nutrition and for growth. So with the benefit of a nanofertilizer, you have a smaller particle that are easily taken up by plants, so their nutrient efficiency is much higher. You can design the nanoparticle to produce a type of smart nanofertilizer, so that the fertilizer may become available to the plant when it actually needs it. So there's a range of different benefits in the field of applying nanotechnology, for example, in agriculture for nanofertilization. But this is still a very young field. Still a lot of research is required in this field. And what is also very important is to analyze and assess the safety and ecotoxicology of using nanofertilizers within the environment. So this is a huge study, still at its starting point, and this is what I'm currently focusing on. So in summary, if we look at the broad overview of our research that we've been involved in thus far, we have tried to address some sustainable development goals. We worked on different types of samples from human origin and from plant origin, and using a diverse set of techniques to answer very important questions for forensics here, in food sciences, food fraud, in biofertilizer field, in agriculture here, with both the biofertilizer, the yeast, and the nanofertilizer here. using nanosciences technology, and also using plants as environmental cleanup agents to accumulate toxic metals from the soil. So I would like to acknowledge a few people. There's too many to mention, but I would just like to sincerely thank my current mentor and head, Prof. Maza, my wonderful colleagues at the lab I'm currently based at, and all my collaborators and friends and family that have helped me greatly along this journey. Thank you very much for your attention. I hope you enjoyed, and Prof. Bright will now also provide you with a series of questions for discussions that I hope you will enjoy. And thank you very much. Thank you.
Thank you so much, Prof. Bright. I think despite the hiccups earlier on, we were able to successfully view the video, and it was nice and clear. I think this is quite exciting information. It really gives a broad picture of what the nanotechnologies can do to answer different research questions, to answer different problems that in the past and the current problems. I think Dr. Cloete covered some important issues around the biology, agriculture, medicine, different applications of this type of technologies. She kind of gave a very good introduction to her journey and provided some good advice for early career scientists. One of the issues that she really emphasized on was collaboration. So as we embark on research journeys to really look into our areas of interest, find opportunities to network with the right experts, especially at conferences, making sure that we engage with individuals within our areas of expertise. But also she mentions working in multidisciplinary teams, and in the work that she presented, it was really clear that a multidisciplinary approach is really key to try and solve some of the problems that we are faced with in the real world. Rather than working in silos, usually people get stuck and try to find solution elsewhere. So really working in a multidisciplinary approach is really key to solving some of these issues. There were a couple of techniques that we were introduced to. For those of us who have not been in the nanotechnology space, one of them, which was quite dominant, was the Pixie, the proton-induced X-ray emission technique. And I think she did a great job to explain the processes that are involved in conducting that type of work, and the issues around the specimen collection, how important it is in terms of the product that you get in the end of the day. I probably had a premonition around the hair, so there was really quite strong reference to hair samples, human hair, in the animal research, looking at protection of our domestic animals, safeguarding them, looking at predators, and the use of hair as a sample for that. And it had many applications, looking at the different chemical properties that it has and the advantages of hair as a sample. And one of the areas that she mentions was also forensics and its utility in forensics and as an advantage over other types of samples in the use of nanotechnology. So I think at the end, she really gave a good summary of the different areas that nanotechnology can be really useful, looking at the different problems that we are facing in the world, like climate change, how we can use nanotechnologies to look deeper into the resources that we have in the agriculture, to see, how we can use nanotechnologies to improve on the products that we are producing. And one of them was fertilizer production, coming up with variants that improve on our agricultural sector. She also touched into the food science, looking at coffee, and the different types of coffee from different regions, and then its application in food analysis and looking at the quality. There were aspects that I was also not aware of around food crimes or food fraud, which can be also investigated using this type of technology. So besides the Pixie, she also talked about MEV SIMS, which is secondary ion mass spectrometry, which is another technique that can be used in ion beam technology, and the different aspects of the two techniques and all the studies that they've taken up in her realm of work, which really show the use of nanotechnology in real life to solve real life problems and advance the science and the technologies that we have at hand. So I would like to now refer back to Dr. Nwaru. As we heard from the end of the video session, there are a couple of questions that Dr. Karen shared with us for the group to kind of discuss and look into. So we will have a few minutes for each question, maybe 5 or so minutes in the interest of time, so that we be able to finish within the allocated time. Thank you, Dr. Nwaru. So the first discussion question from Dr. Karen is, since I touched on multidisciplinary in research in my talk, how can one use science diplomacy to promote a multi or interdisciplinary career path?
I mean, if you want to contribute, you can raise your hand, then I will unmute you.
Okay. So I hope we can all see the screen. There is some black patch that is covering the screen. Okay, yes, it's gone. All right, so if you would like to contribute, you can just raise your hand, and Dr. Nwaru will give you the opportunity to speak. I don't know, Dr. Anwaru, from your end, if you can see any hands. I can't see from this end.
No, I don't see any hand here.
Okay. Is the question clear? Since she touched on multidisciplinarity in research in the talk, how can one use science diplomacy to promote a multi or interdisciplinary career path?
Perhaps not many people are doing this or experience in science diplomacy.
Yeah. Okay.
Go to the next. Okay, raise your hand.
Oh, raise your hand. Okay.
Francis, you can unmute yourself and speak.
Good evening, sirs.
Good evening.
I think the last.
Francis, we don't hear you anymore.
Yes, the last comments in the study of, some we are limited in pledging that, and share a brief on that so that we can also know what to contribute to.
Francis, we didn't get your point actually. Your line was not good.
Oh, yes, please. What I said was that, okay, please, can you hear me now?
Hear you now.
Okay. Yes. I think you sort of made a passing comment that it seems most of us are not in the area of science diplomacy, and so we kind of limited in contributing to this discussion section. But if there can be a brief, intro to that aspect of, are we looking at how to actually get in touch with somebody in an area that perhaps you want the person to contribute to your research? How do you go about it? Is that what the diplomacy is all about, or there's kind of a structure that the person perhaps could follow to get to a fellow in an area of expertise that a researcher would want to get on board for a study? I mean, if anybody can brief us on, if the host or any person has a knowledge in science diplomacy, could just throw light on it for us. Thank you.
All right. I see Leah is raising her hand, but I think science diplomacy is an area that is really becoming popular nowadays. I think it's just combination of different research fields and see how they can be used in international affairs to solve national and international developmental challenges. So the combination of different disciplines as well as international affairs and diplomacy, and bringing science to the context to solve national and international affairs. But let me give Leah time to see if she has additional points to make here. Yeah, Leah, please.
Hello, good evening.
Good evening.
Good evening.
So what I want to say is that what I know is that science diplomacy is an umbrella which can just tackle different disciplines like technicals, research-based, etc. So I don't know much on this chemical something, I don't know, but I think, as you said, science diplomacy can handle many disciplines. Thank you.
All right, I have Evans.
Hello.
Hello.
Yeah, I think I missed this presentation, but I have some practical experience with respect to multidisciplinary research and then the diplomacy. So let's say as a young career, a scientist, one of the practical steps is that first you attend a few conferences. So based on the conference resource personnel, the person does the presentation, then you get to know the specialty of that person. So in designing a proposal, you have to call in a lot of expertise. So based on the conferences you attend, you have a pool of research, senior researchers from different disciplines to add up to your team in your proposal to aid you to win grants as a young scientist. Then secondly, your scientific results must be impacted at the society level. So the science findings and the politician, that is where the diplomacy comes in, bringing the expertise from the scientific and the socioeconomics and the policy makers. Those are the politicians. So if you are able to invite politicians, media, non-governmental agencies to your conferences, it aids you to impart your findings to the society, and so therefore policy makers can understand the details of the research, and so help in passing out laws which will go to affect the society positively. That is how I get it.
All right. Do we go to the next, Kevin?
Yes, please. I think, yeah, we have tackled the question. So the next question that was provided from Dr. Karen, what are the perceptions of and attitudes towards multidisciplinary research by students and staff at your university? What are the perceptions of and attitudes towards multidisciplinary research by students and staff at your university? Okay, if you want to contribute, you can still raise your hand.
May I have a comment?
Yes, of course.
Yes, thank you. I think this presentation is an eye-opening for several areas of collaboration. About multidisciplinary research, there are two opposing facts. The first fact that multidisciplinary research participates in development of the society, and also it has a great opportunity for increasing visibility and citation, and also all aspects of impact of research. And this is motivating for researchers to go on this way and engage in multidisciplinary or interdisciplinary research. On the opposite hand, the promotion rules in my country and in several countries in Africa and in the Arab world, as I see, are discouraging in some part of multidisciplinary research, as they give a high number or a high degree for single researches, and the degree is decreased when the number of authors in the same discipline are increased, unless your research is published in Q1 or Q2 journals. So some researchers opt from participating in multidisciplinary or interdisciplinary research because it's no value for their promotion. And so we need a discussion between those policymakers in universities and researchers to get the best value of this. And as I know, we shouldn't do research for promotion, but in fact, researchers need research for their promotion. And that is the fact. And what we need to reach a point of agreement and of motivation to push research for development of the society. Thank you.
All right. Any other addition? Anyone who wants to contribute to that? What are the perceptions of an attitude towards multidisciplinary research by students and staff at your university? Okay, we are aiming that in our May edition, and that is the 4th edition of this seminar series, to dig deeper into multidisciplinary research. Like Eman highlighted, there is a culture that is pervasive across African universities trying to promote single discipline or singularity of research. And that is unfortunate because that is not the way a society is shaped. And that is not the way our society is mirrored or the way our society can grow. Every society grows with intersection, multi-sectoral collaboration. So different disciplines really are required to think together because eventually our research is to influence societal changes. And if we do not achieve that, then our research does not make any sense if it does not impact on the society. And the society is a place that is multi-sectoral in nature. And so different disciplines are required to work together to provide lasting solution. Otherwise, it is difficult and challenging to work in silos. So this should be encouraged, and hopefully all things being equal, we can continue on this discussion and see how others have also implemented this this style strategy in their own research. All right, perhaps I go to the next one, Kevin.
Yes, I think we can move on to the next question. All right, then question 3, how can basic and applied research be used to address the sustainable development goals in Africa and your specific countries? How can basic and applied research be used to address the sustainable development goals in Africa and your specific countries?
Please raise your hand if you have contribution to make on this. Okay, I have Evans.
Hello.
Yeah, we hear you, Evans.
Yeah, I think for basic and applied research should be applied and apply in our individual countries to get a positive impact. There should be a clear-cut definitions and agreement between the universities, the research institutions, and industry. The course design for our universities should be in such a way that all basic sciences which are taught to students don't end up on the shelves, and all research work should be 100% applicable. So therefore, research from the ideas from the basic sciences can be applied through linkage between the universities, the research institutions, and the industries who will be there to uptake the what we call technology development from the combination of the basic science and apply research at the research institutions, and then finally to be implemented based on grants set aside by the individual countries to ensure that basic science does not remain in the academia, but ends up in our society.
All right. Any other? Any other contribution outside what Evans has said? I think just to conclude on this section, from Karen's presentation, one of the things that continue to come out is that there is room for partnership, collaboration, and regardless of the stage of the research trajectory we find ourselves, whether it is basic, whether it is applied. Once we are initiating research and we are thinking of the society as the custodian and the laboratory of application of our research, then we have room to think along with other colleagues in different disciplines. Imagine that many researchers work just in the lab, and they sometimes produce research results that are very relevant. For instance, the COVID-19 situation we find ourselves. Vaccine has been provided and developed. Those who did this vaccine, you have different levels of trials, phase 1, phase 2, phase 3, phase 4. Those who engage in feasibility studies, studying the efficacy and then the effectiveness, cost effectiveness. Many of these people are buried in the lab and do not have anything to do with who gets these vaccines and who don't get it. So what happens is that once they have done this research and they have found things at the lab, there is other group that will take it up and move it to the ladder of taking this to the healthcare settings. There are those who take it also from there and go to the market. There are those who will take it from there to convince the policymakers, the government, the need for these things. There are those who will take it from there and begin to convince population, because you have people who have different level of thinking, who are doubtful of what this vaccine can do. So there is that level of communication. So in essence, if you're doing basic research, you have to know that if you do not have partnership and collaborators and the other aspect of researchers that follow the trajectory of getting this research result to the society, it wouldn't work out. So it's very important for us as Africans and institutions and universities and institutions of research to know that we need to promote multidisciplinarity. We need to promote collaboration. We need to promote partnership. And I think it was Evans who was mentioning partnership with the industry. These things are very important, and we need to be very natural about them, because that is the only way we can move forward to have the check and balances and to ensure that the research we do is producing the right results. All right. So Farai, Kevin, thanks very much. I will take it over from you here to thank you for moderating this session and for everyone that has been present here. As you can see, the topic this evening, it was quite technical to an extent, but it was still very, very simple to understand what Dr. Cloete was trying to pass through, that research has lots of flexibility and beauty, that you can engage in wedding even within research. You can marry different fields. You can tango, you can dance, you can stretch your hands and pull people from different areas, and that is the beauty. And so as African scientists, and as you can see, one of the things we try to do in ASFI in our trainings is to lump you together, to fight it out from enemies to friends, because that is the only way we can go. We need collaboration. We need multidisciplinary endeavor, because without it, it's really difficult to do things in silos. If everything goes well, in our May and 4th edition of the seminar series, we are going to bring this topic back again to the board, because we want really to intensify this discussion. How do we ensure that we are working together? Because everybody needs to put their hands on the deck to get this work, and especially looking at where our continent is. So thank you for coming this evening. I hope you can take one thing back home, and then wish you a very lovely week ahead. Eman, do you want to take picture, or you want to let them go?
Yes, we can take picture, but
the picture
before
okay.
we leave, I want to say that the discussion is not ending. We appreciate if you have comments, you can leave it under the video on YouTube, and we are happy to respond, and also we will circulate to Kevin to respond, and the discussion is going on.
All right, you can share your video and you can unmute yourself, and let Eman take the picture of the happy faces.
Thank you.
Okay.
All right. Thank you very much, everyone. Have a good evening. I'll see you again next time.
Thank you all.
Bye.
Bye.
Bye-bye.
Bye.
Bye, and thanks, Prof.
Bye.
Thank you.
Bye. Thank you all.